going to join you a little bit later on today. This is chapter 27, and what we'll be doing is reviewing nutrition and metabolism. So, when you eat, every molecule that you put into your mouth in any kind of food stuff that you're interested in consuming is made up of one of six major classes of molecules or combinations of these molecules. And these include carbohydrates, lipids, proteins, water, minerals, and also vitamins. Now, those compounds with the highest energy content include the carbs, the lipids, and the proteins, with the lipids having the most calories per gram, so that's the most energy you can consume in the minimal amount of grams. The carbs running second place, and the proteins running around third place. The water, the minerals, and the vitamins have little to no calorie content, but they're still important to the normal functioning of the human body because, as we'll see, especially in the case of vitamins and minerals, these serve as cofactors or coenzymes that allow chemical reactions in the body that are catalyzed by enzymes to happen more efficiently. And in the case of water, what's being provided here is a reaction medium in which all these things can take place. So what you're going to see on the next page is something called the My Pyramid. Now this has been replaced by something called My Plate, but essentially this was designed when it was initially introduced to provide for you a guideline as to a balanced diet. It's a personalized approach to healthy food activities and based on gender, age, and activity. Food from each of 6 bands is needed each day. An example would be a healthy, moderately active 18-year-old female requires about 2 kilocalories per day, while a male requires about 2.8 kilocalories a day. The steps are a reminder of the amount of daily activity that's needed. The reason for the discrepancy between female and male calorie requirements is because males generally have a larger concentration of testosterone in their physiology than women do, and this tends to produce more muscle mass, and it increases hematopoiesis, and as a result the demand for calories is higher. Now, as we move on to a look at what constitutes healthy eating, 4 calories per gram for proteins or carbohydrates can be used as a guide for how many calories you should consume versus how many calories that you're going to be burning as a result of your normal activity. 9 calories per gram for lipids. Now, what you'll notice here that the calories are basically a direct measure of the energy content of these molecules. So something that's going to have 4 calories per gram is going to have a lower energy content than something that has 9 calories per gram. And you might ask, well, where is it in the molecule that this energy is hidden? And the answer is, it's in the carbon-hydrogen bonds. So once we ingest these components, they absorb across the GI and they get into the blood, are then transported to the cells, and the cells will then take them up and try and split those carbon-hydrogen bonds in a process known as oxidation. In lipids, you have a lot of carbon-hydrogen bonds, probably 50 to 100-fold more than you have in the typical protein or carbohydrate that's near the same molecular weight. This is why if you eat a diet that's high in fat, you tend to gain a lot of weight, as opposed to a diet that's high in protein or carbs. You'll still gain weight, but you won't gain nearly the weight you would gain with an extremely high fat diet. So what are some expert suggested dietary calorie intakes? We should get 50 to 60% of our calories from carbs, and there should be less than 15% coming from monosaccharides, and less than 30% coming from fats, specifically triglycerides. And of that, less than 10% should come from saturated fat. 12 to 15% should come from protein. And while we're on this slide, let's note these percentages and why they're important. 50 to 60% of our calories should come from carbs. Well, what they're telling you here is that you should try and tilt your energy consumption away from fat and towards carbs, because carbs are more easily oxidized and the energy is more readily available for the body. But less than 15% of these should be monosaccharides. Well, monosaccharides are single sugar units, and the body can very easily take monosaccharides up from the blood and transport them into the cell, and the result is a readily available source for the production of ATP. In complex carbohydrates, we're talking about sugar chains that are more than 2 or 3 sugars long, and that takes more work for the body to transport from the GI tract into the blood, and then once it reaches the liver generally will store this and won't pass it on to the other cells in the body directly. It'll probably end up causing these complex carbs to remain in the body as stored fat or stored glycogen, and then when the demand need comes around, if the cells that are metabolically active require a fuel source, the liver will then mobilize those stored fats and those stored glycogen molecules into either free fatty acids or, in the case of glycogen, free glucose in the blood. And that'll be then transported to the target tissues, who can then immediately use it for energy. Now, less than 30% should be from triglycerides. And remember that fats have the highest amount of calories per gram. And of that, less than 10% should be from saturated fat. Now, why is this? Well, saturated fat represents a molecule that has a very high calorie per gram ratio, and probably more calories per gram in saturated fats than anything else you'll eat on a daily basis. And this is just simply a way to allow your body to cut down on total calories by avoiding these saturated fats. Saturated, by the way, refers to the number of carbon-hydrogen bonds in the lipid that we're talking about. In saturated fats, the number of carbon-hydrogen bonds is at a maximum, which means that the energy content is at a maximum. And in unsaturated fats, we have less than the theoretical maximum number of carbon-hydrogen bonds, and that cuts down on the number of available calories in that compound. So these are just basically calorie guidelines that we're looking at here. Now, 12 to 15% should be from protein. You can take in more protein. It's not going to be terribly harmful. Protein, remember, is made up of building blocks called amino acids, and there's a consequence to the ingestion of protein, which it necessitates the production of ammonia and urea as byproducts. And the only reason that they caution you from eating a diet too high in protein is that it could eventually put a strain on the kidneys and on the liver, because they have to detoxify these protein breakdown products and excrete them from the body so that they don't build up to toxic levels and end up killing us. Two examples of conditions that result from the buildup of nitrogenous waste in the body include urea and free ammonia. If either of these accumulate in the body, you can affect enzyme activity and cause proteins to denature, and the result will be a cessation of chemical reactions that help life to proceed. So this is why the restrictions on protein, why we can't be all protein all the time. So we should eat a variety of foods in order to balance our intake of these three major food groups: the carbs, the proteins, and the fats. We should also maintain a healthy weight. Now, the reason for this is several-fold. One is the fact that in saturated fats, in individuals who don't maintain a healthy weight, what happens is that they accumulate triglycerides, which are a form of saturated fat. Now, in the event that you begin to gain what we call bad weight, that would be adipose tissue, you can end up compromising the activity of several body systems at once. One would be the circulatory system. As you gain unhealthy weight, what happens is that your blood vessels, instead of becoming more numerous, become longer. And when that happens, you increase the resistance against the beating action of the heart. What that does is two things. It produces hypertension, but it also can produce cardiomyopathy. Other dangers that go along with this are the fact that high pressures in capillary beds can cause them to burst. And this can result, for instance, in the retinas of the eyes being damaged because they are perfused by capillary beds, or the loss of extremities because they are also perfused primarily by capillary blood feedback. We should choose foods that are low in fat, saturated fat, and cholesterol. And the reason for this is this cuts our calories. It also cuts the amount of bad cholesterol in the blood. If this bad cholesterol gets too high, examples of bad cholesterol being things like low-density lipoproteins or very low-density lipoproteins, or in extreme cases, chylomicrons, in the event that these compounds accrue, what can happen is they can deposit on the walls of arteries producing plaque, and this causes the arteries to stiffen, causes the lumens to narrow, and causes the resulting blood pressure to go up. And the danger here is that the high blood pressure could do several things. It could, again, cause capillary beds to burst, but more importantly, it could potentially knock free some of these plaques on the walls of these blood vessels and produce an embolus, which could become lodged in the capillary bed and choke off oxygen and nutrients to the tissues that are downstream. We should eat plenty of fruits, vegetables, and grains. Again, this ups our carb intake, but it also ups our intake of phytochemicals and vitamins. A lot of plants are very rich in the vitamins that we need in order to maintain life. Things like vitamin C and vitamin A are abundant in certain types of vegetables and leafy greens. We should use sugars in moderation. Now, when we say sugar, we mean not just good old table sugar like that you would see in a bag of Domino's sugar, but we also mean the ingestion of starch. Now, starch is a type of sugar. It is a polysaccharide, and in the case of plant starch, what you're really looking at are chains of glucose molecules hooked one to another. And one of the first things that happens when these compounds enter our body as a result of our ingesting them is that they get broken down into monosaccharides. And so in terms of how the body deals with it, it's kind of the same thing as eating just straight monosaccharides in the same molar amount. You might get a little bit less of an insulin spike, but you're still going to end up in the long run probably gaining unhealthy weight as a result of concentrating on these things. Examples of foods that have a lot of starch in them: potatoes, pasta, rice, breads. You can probably think of all manner of products that are high in starch content. We should use sodium in moderation, less than 2.3 grams per day. Sodium is a major electrolyte in the body, and it controls fluid movement. And in the event that we retain too much sodium, what can happen is that it can affect now the body's total fluid content, and that could throw cells out of homeostasis. We should also use alcoholic beverages in moderation. Two reasons for this. Number one, believe it or not, alcoholic beverages are high in calories, and that all by itself is not good for you. But in addition to this, alcohol has to be cleaned by the liver using an enzyme called alcohol dehydrogenase. And one of the intermediates in the detoxification of alcohol is the production of a molecule that has properties very similar to formaldehyde, which is a tissue fixative. And if the steady state levels of this molecule build up too high, what it can literally do is pickle the liver and lead to something called cirrhosis of the liver. Recommended amounts are 1 drink a day for women, 2 drinks a day for men. So, the main categories of nutrients include carbs, proteins, and lipids. Highest calorie content, again lipids, second most carbs, and bringing up the rear are the proteins. There are calories in each of these for energy, and they also can serve as components of building materials. Water and minerals are also necessary. Some of the minerals include calcium, phosphorus, potassium, sulfur, sodium chloride, fluoride, so forth. And there's a table in your book, 21.1, that lists all of these. And one of the things that you might notice is that in these minerals, they may not be present in very high amounts. So does that mean they're unimportant? And the answer is absolutely not. For instance, we rely very much on iron, even though it's only a trace mineral in our body when compared to things like carbon, oxygen, and hydrogen. Yet if you didn't have any iron in your body, you wouldn't be able to carry oxygen in the hemoglobin that's inside your red blood cells, and as a result, you would end up dying. So these are the kind of roles that these minerals play, and that are often trace elements. If you take these minerals out of the diet, you have a mineral deficiency, and it's going to negatively affect your health. It'll put you in a state of disease or disequilibrium. Vitamins are also essential. You can get these directly, or you can consume dietary provitamins that your body then can convert into the active form of the vitamin. Examples of lipid-soluble vitamins are A, D, E, and K, and the water-soluble vitamins are B and C. Many of them function as what are called coenzymes. The best way to think of coenzymes is that they serve as molecular tools for the body's enzyme catalysts, and so that allows you to produce needed products for the tissues of the body as a result of these continuing activities. Without these coenzymes, a lot of enzymes would be unable to perform their normal job. The reactions they catalyze would slow down or stop, and all those other processes that are dependent on that particular chemical product that arises from that chemical reaction would suffer. They'd be left out in the cold. Okay, metabolism is facilitated by enzymes and coenzymes. An example of a coenzyme is NAD, which comes from niacin, and FAD that comes from riboflavin. These both act as electron carriers. They're important in a bodily system known as the electron transport chain. And the reason the electron transport chain is a big deal is because high energy electrons that are fed into this chain, which exists basically inside the inner membrane of the mitochondria, become oxidized over time. And this causes energy to be converted from one chemical form to another. The ultimate chemical form of energy is ATP, which is the biological gas in the tank that the cell absolutely has to have in order to survive. So these coenzymes are vital. If you didn't have sufficient amounts of riboflavin and niacin in your diet, again, you would have a problem generating sufficient amounts of ATP to run the body. A sequence of enzymatic steps is known as a pathway. There's two categories: anabolism, which is the synthesis of more complete chemicals, and catabolism, which is the breakdown of a certain molecule with the subsequent release of energy. 40% of our cellular functions rely on ATP. 60% of these chemical reactions generate excess heat. And while we're on the topic of ATP, what we should note is that the highest energy bonds in this particular molecule exist between the phosphate groups, because the phosphate groups contain a negative charge on them, and it's very difficult to keep two negative charges together unless you have this sort of special safeguard. Okay, what you're looking at here is just an overview of metabolism, and it's designed to illustrate the energy flow between the molecules that you consume in order to extract the energy from the food that you eat, and how that energy is used to keep the human body going. So here we can start over with complex molecules, glycogen, proteins, and triglycerides. And where do those come from? Well, they can either be stored as energy reservoirs in the body, or you can consume them in the food that you eat. Regardless of how you acquire them, these can be broken down into smaller components, and as those bonds are split, the energy from those chemical bonds does two things. It releases heat as a byproduct, and it generates now simple molecules such as glucose, amino acids, glycerol, and fatty acids, which can be used in anabolic reactions to synthesize more complex molecules if we're using their structural components, or we can use the catabolic reactions whose energy that we extract as a result of cleaving high energy bonds, the carbon-hydrogen bonds, to generate ATP. And ATP is used to drive energetically unfavorable reactions that otherwise wouldn't spontaneously occur. This is, remember, biological gas in the tank. This is the type of energy that the body can readily understand and utilize. No processing is required. The energy from this high energy molecule is transferred to anabolic processes. The result is, again, the release of heat and also the synthesis of complex molecules such as glycogens, proteins, and triglycerides, or we could say the production of work as well. Okay, so we should put work in here sort of as another little arrow branching off from this green box. Okay, so we release heat in any event, we can perform work, or we can synthesize complex molecules that we can then either store or break down. So in general, when we talk about catabolism, we're talking about the breakdown of large molecules, generally with the intent of extracting energy from them, and anabolic reactions are a series of chemical reactions that are designed to utilize a separate energy source in order to build complex molecules. Now, if we look at a diagram showing you some of the major metabolic pathways, what you're looking at here in the white area of this slide is what we would consider to be the cell cytoplasm. And this large sort of cylindrical thing that looks like a fat sausage is the mitochondria. The mitochondria, remember, is nicknamed the powerhouse of the cell. And what this illustration is designed to show you is that in the cytoplasm, what happens is that glucose is split into 2,3 carbon molecules called pyruvic acid. And during the course of this series of reactions called glycolysis, we generate a net gain of 2 ATP molecules, which can immediately be used for cellular energy. However, that's not nearly the total potential number of ATP molecules we can generate per molecule of glucose. That payoff isn't realized until we completely oxidize the rest of the carbon-hydrogen bonds. Now notice that we also produce a molecule called NADH as a result of glycolysis, and NADH represents a storehouse of high-energy electrons. Basically, it's a carrier between the process of glycolysis, in this case, and the process of oxidation of these high-energy compounds to their counterparts. In the case of NADH, it would be NAD. In the case of FADH2, shown down here, it would be FAD. And in the course of that oxidation, that extraction of energy, that energy is then transferred ultimately to ATP synthase. Generates now straight ATP in the electron transport chain as the result of the establishment of a chemiosmotic gradient as these electrons go from an area of high energy to an area of low energy. But we'll break this down step by step. Let's just start at the top with glycolysis. So the sugar, the 6-carbon sugar, is split into 2 3-carbon sugars, and the byproducts here are ATP, which can immediately be used for energy, and NADH, which we'll rejoin here at a later date. Now, in the presence of oxygen, what can happen is that 2 more branches of metabolism are relatively active, those being the Krebs cycle, which takes place in the mitochondrial matrix, and the electron transport chain, which occurs on the inner mitochondrial membrane. So let's start with the Krebs cycle. What is the Krebs cycle? The Krebs cycle is essentially a series of biochemical reactions which takes in acetyl groups which are produced as a result of the transport of pyruvic acid into the mitochondria, and then it joins them to a molecule of oxaloacetate, and it forms the intermediate citric acid, hence the other nickname for the Krebs cycle, the citric acid cycle. What then happens as this citric acid moves through the next few biochemical transformations is that CO2 molecules are stripped off, and that reaction releases sufficient amounts of energy to produce either ATP directly or high energy molecules such as NADH and FADH2. Remember, we also produce a few of those up here in glycolysis. The carbon dioxide eventually is carried away by the blood and exhaled as we breathe out. But what happens now to these high energy molecules that we produced as a result of up here these first stages of glycolysis and the Krebs cycle? Well, they donate their high energy electrons to a series of enzymes that are embedded in the intermitochondrial membrane that make up a complex called the electron transport chain. And the electron transport chain does exactly what you think it does. It transports electrons from one physical carrier in the membrane to the next. When it does this, as these electrons move from a high energy carrier to a lower energy carrier, that energy is trapped and used to pump protons from the mitochondrial matrix into the space between the outer mitochondrial membrane here and the inner mitochondrial membrane there. And that produces something called a chemiosmotic gradient. Basically, you build protons up in this intermembrane space. The electrons continue to flow down this electron transport chain from carrier to carrier until they reach the terminal electron acceptor. Well, what is that? That's the last stop in the chain. At this point, these low energy electrons are donated to oxygen, and those electrons plus the oxygen plus a couple of protons make a molecule of water. And where does the water go? Out into the cytoplasm, and ultimately it's carried away by the bloodstream. So this is what we term metabolic water. So two of the end products of metabolism we've seen here, H2O, water, and CO2, which is the waste gas. Now, what about this chemiosmotic gradient that we established? Well, what happens with this buildup of protons in this intermembrane space is that they eventually diffuse back from the intermembrane space into the mitochondrial matrix through a very special, funky-looking, mushroom-shaped protein complex that's embedded in the intermitochondrial membrane called ATP synthase. And what happens as these protons go from an area of high concentration to an area of low concentration is that kinetic energy is trapped by the ATP synthase and used to synthesize ATP from ADP and inorganic phosphate. And that right there is the major energy payoff that we got from ultimately oxidizing this glucose molecule. It's these about 32 to 34 ATP that we generate per molecule of glucose as a result of the activity of the electron transport chain that really pays the energy debt for the body. Now, where does the ATP go once it's made? Well, some of it stays in the mitochondria in order for the mitochondria to continue to perform biosynthetic activities, because believe it or not, the mitochondria is, in a sense, a living entity all its own. It has its own DNA and its own ribosomes and its own set of organelles. It is very similar in some sense to a bacteria, except that it cannot live independently of the host cell. The rest of the ATP flows out into the cytoplasm, where it's used by enzymes that perform anabolic processes to build big molecules or to do work. What would be an example of ATP used to do work? Muscle contraction. Okay, without ATP, we cannot have myosin and actin interact and generate those cross bridges and allow the muscle fibers to shorten, and that's what produces the force of motion. All right, that's where it ultimately comes from. How about an example of an anabolic property that's powered by ATP? Well, that could be something as simple as the biosynthesis of protein, okay, which is powered by ribosomes and the use of transfer RNA and messenger RNA as a template. But this biosynthesis would not happen were it not, again, for the uphill power that's provided by ATP that drives that reaction forward. ATP is also used to synthesize sugars in the body in a process known as gluconeogenesis, and it's also used to generate stores of fat in the body, and this is lipogenesis. And where do these anabolic molecules go? Well, they go wherever we need them. They can be stored as fuel for later use, or they can be structural components of other macromolecules inside the living system. So this is how we go from stripping energy out of a fuel molecule turning it into an intermediate energy store, and then ultimately sucking the energy out of those high energy molecules and using them to generate ATP, which we can immediately utilize to do work. The reason that we have to do this is that we cannot just simply store massive amounts of ATP inside the cell. The reason for that being that it would have to be hydrated, it would take up a lot of physical space, and more importantly, it's extremely easy to degrade. ATP breaks down very easily, so it's not a reliable long-term source of energy storage. So we sort of go to the pantry and burn a little fuel to make this quick energy intermediate that we then use right away. And then what happens when we need it again is we simply resynthesize it. Right? So that's the reason for the energy flow that we see here. Okay, glucose is the body's preferred source for ATP production. It's very easy to oxidize glucose. You don't need as many biosynthetic or biocatalytic steps to break it down and get the ATP payoff. Possible uses include immediate use for ATP production or the storage as glycogen in liver or skeletal muscle for use later on. The leftover glucose that we don't store as glycogen or immediately use for ATP, we store as triglycerides. Basically, we take the energy that's in the carbon-hydrogen bonds in glucose, and we use that to drive the biosynthesis of triglycerides, which we can then store in the body as fat. That's what our adipose tissue contains large amounts of. It enters the cell by facilitated diffusion. Basically, that means there's a protein carrier in the membrane that provides the perfect hole through which the glucose can slide in order to gain entrance to the cytoplasm. And insulin actually increases the rate of transport. So insulin basically is the sugar message. When there's sugar in the blood, insulin production goes up, and that increases the rate of transport of glucose into cells that either need it for energy or into cells that can utilize it to biosynthesize either glycogen or fat. The overall breakdown of glucose is shown here. Basically, glucose and oxygen combine to generate 36 to 38 ATP molecules, with the byproducts being CO2, which we exhale, and water, which we lose either as sweat or in the urine stream. Glycolysis occurs in the cytosol, where glucose is converted into two molecules of pyruvate, with ATP and NADH being byproducts. Now, I should note here that these are not stoichiometrically correct equations. You actually get two pyruvates for every glucose that you break down, and you get two ATPs and two NADHs and two protons for every glucose that you break down, because you end up with two 3-carbon molecules at the end of the day when you split sugar. That's what glycolysis means, to split sugar. This is called anaerobic cellular respiration because it can occur independently of the presence of oxygen. The transition step in mitochondria is where pyruvate crosses that outer mitochondrial membrane and enters the mitochondrial matrix. And part of the energy that does that is provided by the stripping off of a CO2 molecule from the pyruvate molecule, and you leave behind now an acetyl group, which is then hooked to a molecule called coenzyme A, which acts as sort of a big adapter arm that allows this high energy 2-carbon group to ultimately be transferred to a molecule of oxaloacetate, which is one of the first intermediates that initiates now the Krebs cycle, which will be coming up here in a minute. And again, you get two pyruvates for every glucose molecule, so you can go ahead and multiply these guys through by 2 to get an accurate picture of the energy payoff. The Krebs cycle occurs in the mitochondria. Now, it lists here that it is aerobic in nature. It produces as its byproducts CO2 and protons, but it also generates an immediate ATP payoff for each crank of the cycle. You do directly generate some ATP, but the bulk of the energy is actually temporarily stored in molecules of NADH and FADH2. Now, you might ask, well, wait a minute here. You said it's aerobic and oxygen dependent, so where is the oxygen in all this? Well, the oxygen actually comes in when we take these high energy molecules and oxidize them into their counterparts, the oxidized counterpart of NADH being NAD and of FADH2 being FAD. And there, the only reason we're able to oxidize these reduced molecules is because ultimately of the presence of oxygen at the end of the electron transport chain. So it's really the electron transport chain that is directly oxygen dependent. If we didn't have oxygen around, you could probably get some pyruvate into the mitochondria, and you could probably crack it with a few turns of the cycle, but eventually what would happen is that these byproducts would build up and the cycle would stop because we're not able now to drain these end products off and favor now the forward reaction of the cycle. The electron transport chain is directly aerobic. It is dependent absolutely on oxygen to function. And here you can see some of the overall equations. NADH and FADH2 enter the electron transport chain at actually different points. NADH actually donates electrons of the highest energy to the electron transport chain. FADH2 donates electrons at a slightly lower energy to the electron transport chain. So you get a little bit fewer ATP per FAD than you do NADH, but it still results in a big energy payoff. You combine this with oxygen, which sits at the end of the electron transport chain and accepts those energy poor electrons to form water, which you can see here, and again, tons and tons of ATP, between 32 and 34 molecules per every molecule of glucose that we burn up, that we oxidize. Something to sort of keep in mind when we're talking about all this metabolism. An oxidized molecule you could think of as an energy poor molecule, whereas a reduced molecule you could think of as an energy rich molecule. So what are some examples of oxidized molecules? Well, molecules with very few carbon-hydrogen bonds in living systems. What would be a reduced molecule? Molecules with a lot of carbon-hydrogen bonds. Remember that the carbon-hydrogen bond is the most energetic of the bonds in the food that you eat and in the macromolecules you consume. Okay, so how do we regulate blood glucose in all this? Basically, when you eat, what happens is that we break down the macromolecules into smaller and smaller pieces through enzymatic activity and also mechanical breakup. And we absorb the monomers into the bloodstream. The monomers of proteins are called amino acids. The monomers of polysaccharides are called monosaccharides. And it turns out that lipids are actually transported in the blood in the form of little vesicles, which can take on various names. Some are called chylomicrons, some are called VLDLs, some are called LDLs, and some are called HDLs, and that stands for very low density lipoproteins, high density lipoproteins, and very high density lipoproteins. This is just acronyms. And those components are transported ultimately to the liver. And what the liver decides to do is to store them or pass them on in the blood to the rest of the body. So when blood glucose is high, what happens is that glucose in the presence of ATP is used to synthesize glycogen, a macromolecule which is then stored in liver and muscle. Now you might ask, well, why can't we just store all our energy in the form of glycogen? And it's a good question. And the answer is that you don't get enough bang for your buck in terms of storing your body's energy reserves as glycogen, because glycogen is not as energy rich a molecule as fat is. The effect here is that blood glucose level drops in the presence of, now the signal here being insulin. When blood glucose is low, what happens is that the hormone glucagon is produced by the pancreas, and it enters the bloodstream, and it triggers now glycogenolysis, which is the breakdown of glycogen into glucose molecules, and also a process known as gluconeogenesis, which is an ATP-dependent biosynthesis of glucose in the body. Glycogen can be broken down to glucose, and hormones such as glucagon and cortisol stimulate gluconeogenesis in the liver cells by breaking down glycerol from fat and using that to form glucose, and also transforming certain amino acids into glucose. Okay, so this is not an energy efficient process because we consume ATP to do it, But if you think in terms of total energy balance, you've got so much chemical potential energy stored in your fat that we can afford to lose a little bit in this inefficient process. So this represents, again, a huge store of potential energy. Fat, since it has so many calories per gram and is hydrophobic, takes up less physical volume than something like glycogen would. And so it represents an extremely efficient way to store large amounts of energy. And so that's probably why we were selected to store lots and lots of fat in our body tissues, one of the reasons anyway. And this is just looking at the interplay between blood glucose and the body's energy needs here. So let's start over here on the right. Triglycerides basically are molecules that are made up of glycerol, which is an alcohol that contains 3 hydroxyl groups and is coupled to 3 fatty acids. Now, fatty acids, if you remember the structure of phospholipids, phospholipids contain fatty acid tails, very long hydrocarbon tails, and that's really where most of the energy is in the triglyceride. Triglycerides can be broken into glycerol and into fatty acids. Where does the glycerol go? Well, the glycerol can be converted into a special intermediate called glyceraldehyde 3-phosphate, which is actually one of the intermediate steps in glycolysis. As a result, it can be used to generate glucose, okay? In the reverse, all right, it could be used to generate ATP if we go in the opposite direction. Now, I should point out that the molecules that are responsible for the biosynthesis of glucose are distinct from the molecules that are responsible for the breakdown of glucose, and they're actually partitioned in different compartments in the cell and in different tissues in the body. Okay, so it's not like the same enzyme is doing 2 different tricks. It's 2 sets of enzymes, but the intermediates are very familiar. Okay, the intermediate steps are identical in many cases, the chemical intermediates. Okay, so what happens here? Well, we can biosynthesize glucose, and that can be converted into glycogen, which can be stored in liver or muscle. How about pyruvic acid? Well, glyceraldehyde can be converted into pyruvic acid through oxidation, which will produce now an energy payoff in the form of NADH. And the pyruvic acid can be used to synthesize certain amino acids. We also note here that lactic acid and certain amino acids can be converted directly into pyruvate, which can then be used to synthesize glucose, which can ultimately be stored as glycogen as well. So you can see here that there's sort of a way to get from A to B almost from anywhere in this biosynthetic pathway. Now you might ask, well, wait a minute here. What about these fatty acids? Where do they come into play? And where they come into play is in catabolism. Fatty acids can be broken down 2 carbon atoms at a time, and those carbon atoms can be used to generate acetyl-CoA in the mitochondria, which can then enter the Krebs cycle directly. And so here you can see why there's so darn much chemical potential energy in fatty acids, because the typical fatty acid chain is between 17 and 25 carbons long. And so if you think that for every single turn of the Krebs cycle you're getting an NADH and an FADH2 and an ATP directly, and that for every NADH and FADH2 you're getting about 5 ATP molecules in addition to the ATP you directly generate, so call it 6. You can see that the amount of ATP in, say, a 25 carbon fatty acid is enormous, and so that represents a huge amount of stored energy. Okay, so let's talk a little bit more about lipid metabolism. Lipolysis is where we split the triglycerides that we store as fat in our body into glycerol and fatty acids. Glycerol can be used in glycolysis or in gluconeogenesis, while the fatty acids can be used to produce ATP as a result of entering the Krebs cycle, as we just described. The liver converts some acetyl-CoA into ketone bodies, and that generally happens when we're running a little low on carbohydrates in the diet. What's the best way to think about this? There's an old saying that I remember from my biochemistry text, which states that fats burn in the flame of carbohydrates. And what does that mean? It means that you need a lot of those Krebs cycle intermediate compounds like oxaloacetate, citric acid, and isocitrate, and so forth, in order to fully oxidize now these 2-carbon units. In the event that you don't have enough of that in the mitochondria, what happens is that the mitochondria will sort of go into an emergency backup mode where it will incompletely combust these acetyl-CoA 2-carbon units and form as a byproduct ketone bodies. And that can cause you to enter a metabolic state called ketoacidosis, because these ketone bodies in many cases are biological acids, and what they do is they lower the pH of body fluids, and ultimately that can be harmful. Examples of diets that promote this are things such as the Atkins diet or the South Beach diet, where we are told to cut back on carbohydrates while only moderately cutting back on our fat intake. And what this does is it incompletely combusts our fat stores, and we lose a lot of that chemical potential energy as volatile ketone bodies. The breath, for instance, of people that are in ketoacidosis often smells like nail polish remover. Diabetics can form excessive ketone bodies, and the reason for this is that they can't utilize efficiently the glucose that's in their blood because the insulin signal, for one reason or another, isn't properly working. Either the insulin isn't being made in the first place, or the insulin signal, even though it's being produced, isn't being responded to by the tissues that would normally act on it. And so what happens here is that these acidotic byproducts build up in the blood and cause this sweet breath, and it can eventually affect the pH of your body fluids. This can lead, as it says, to coma and death. Lipogenesis is the synthesis, the biosynthesis of triglycerides. We can take excess fat, carbs, or protein, and we can convert them into fat and store them in the body, and this just shows you how that occurs. Okay, so we can start up here with glucose, which we know can be split into pyruvic acid through the process of glycolysis. But what can happen when we're at the pyruvic acid cycle? We can generate acetyl coenzyme A. And what we can then do with that acetyl coenzyme A is directly use these 2-carbon units to synthesize 2 carbon units at a time, the fatty acid component of triglycerides. What about up here, where we see glyceraldehyde 3-phosphate as one of the intermediates? Well, this can be converted to glycerol, which can serve as the backbone for fatty acids, and we can synthesize now triglycerides. If you want to see a picture of fatty acids, or of triglycerides rather, go to the index in your book under T and find a page where it shows you a picture, and what you'll see are those 3 fatty acid tails linked to the triglyceride. This is actually a type of molecule known as an ester. Okay. Now, down here, coenzyme A can be used to form fatty acids. It can also form ketone bodies, which we know are not a desirable byproduct. Also note that certain amino acids can be converted into acetyl coenzyme A. Some can be converted into pyruvate, and this allows them to enter the Krebs cycle and or glycolysis and be used directly to generate energy. So this is why when we consume protein, we don't just get biosynthetic building blocks from the amino acids, we also get a direct energy payoff. In fact, it takes the body more work, more biochemical work, to completely process protein than it does to process carbohydrates or fats. And so one of the prevailing theories here is that if you eat a lot of protein, what you do is you produce a lot of heat as a byproduct, and a lot of that doesn't end up as biosynthetic molecules in the body, and it gets lost as heat. And so this can help you lose weight. It's better to try and consume a higher protein diet in lieu of, say, eating simple sugars or starch, for instance. Okay. How do we transport lipids in the blood? Well, they're not water soluble, right? They're greasy, so they need a special vehicle in which to be driven from point A to point B in the body. And this protein coating is called a lipoprotein. Basically, it's a combination of lipids and proteins together in like a little hollow bubble, and it gets carried in the bloodstream to and from the liver and to and from muscle, to and from cells that need it for energy and for biosynthesis purposes. The types of proteins include chylomicrons, which carry dietary fat straight to the adipose tissue. So when you have high chylomicrons in the blood, that means you're in a lipid storage mode. And very low density lipoproteins, which are synthesized in the liver, and they transport fat directly to adipose tissue as well. So the VLDLs and the chylomicrons would be considered what we call bad cholesterol. These can be converted to low density lipoproteins, which are also considered bad cholesterol. They carry about 75% of the cholesterol to cells for use there. High-density lipoproteins are considered good cholesterol. And the reason for this is that they are indicative of the removal of excess cholesterol and the transport to the liver for ultimate removal from the body. Okay, so when you have a lot of HDLs or VHDLs and very few of these bad cholesterols, what that means is that you're in a lipid burning or losing mode, whereas if you're high in these bad cholesterols, that means that you're very much in a lipid storage mode. So this is the difference between good and bad cholesterol. Now, there's another consequence to this, which is that if you have a lot of these bad cholesterol and lipoproteins in your blood, what happens is that they can produce plaque on the insides of arteries, and that can cause the artery lumens to constrict, and that results in hypertension. And that's bad because that can ultimately result in burst capillary beds, say in the brain or in the heart. Or it can also lead to basically hypertrophy of the heart, because you have to work very hard to push the blood through these very narrow openings. Okay, so you got hypertension, hypertrophy, and the potential for embolism, all as part of really the same disease process. Okay, so how do we deal with protein? Well, protein breakdown has an important aspect to it, which is that proteins contain nitrogen, and nitrogen balance is vitally important for the body. This is why the building blocks of protein are called amino acids. The amino component is the nitrogen part of the amino acid, and the acid part is the carboxylic end, which is at the opposite end of the monomer. So catabolism is the breakdown of protein. It is deaminated in the liver to remove the amino group and form ammonia. Now, on the face of that, that's good, because we can now use that resulting deaminated amino acid either directly for energy, or we could store it as fuel, okay? Or we could, say, convert it into a completely different amino acid down the road. But there's a problem here, which is that we've got this ammonia, this free ammonia in the system, and we have to get rid of that because that's toxic. The liver converts this into urea, which does so by combining it with a CO2 molecule, and that ultimately gets excreted in the urine. So that's how we keep our nitrogen balance at a steady state. Generally speaking, you want to be taking in as much nitrogen as you're losing. If you're taking in more nitrogen than you're losing, that could ultimately result in nitrogen toxicity. Diseases that result from that include azotemia and uremia, and that can ultimately lead to coma and death. If you are consuming you're consuming less nitrogen than you're excreting, what that means is that you may ultimately run out of building blocks, okay? And so you won't be able to engage in biosynthesis of protein. The rest of the amino acid can enter the Krebs cycle and can be used to make ATP. In anabolism, protein synthesis is used to make a lot of different proteins in the body. You've got 10 amino acids you have to eat in order to have them in your system, and those are called essential amino acids. The non-essentials your body can manufacture from other compounds, so we don't have to eat them to get them. Heat is a form of energy. It's a form of energy that, in a sense, is not usable. We produce heat and it's lost to the universe, and that actually contributes to the further randomization of our environment. It is measured in calories. 1 calorie is considered the energy needed to raise 1 gram of water 1 degree centigrade. So that's the scientific definition of calorie. It's a measurement of heat. A measurement of heat. Now, I'll tell you something about water, which you may or may not know, which is that water has a density of 1 gram per milliliter. All right? 1 gram per milliliter of water is the density, so 100 milliliters of water is going to weigh 100 grams. Also, there's another name for a milliliter of water. That's a cubic centimeter. Cubic centimeter is sometimes abbreviated cc. You've probably heard it in clinical environments where they say give this person 10 cc's of saline, for instance. That's 10 milliliters of saline. For foods, we don't use the calorie because it's too tiny of a measurement. We use the kilocalorie as an abbreviation for calorie with a capital C. So when you see calorie with a capital C, we're referring to 1,000 of these chemical calories, the amount of heat needed to raise 1 gram of water 1 degree centigrade. Heat is generated as a result of the chemical breakdown of the food that we consume, and the rate of heat production is termed our metabolic rate. Your basal metabolic rate is the heat that's produced from catabolism when you're resting or when you're fasting. The basal metabolic rate for an adult is usually between 1,200 and 1,800 capital C calories per day. Okay, so how many little c calories would that be? Well, that would be a 12 with 5 zeros after it. Okay, so 120,000, right? 120,000 little calories per day. All right, between 120,000 to 180,000. Okay, your metabolic rate is affected by exercise, which can increase the rate up to 20 times, or hormones, where thyroid hormone is a potent accelerator of the basal metabolic rate. Your nervous system can also increase your basal metabolic rate, and warming can increase your basal metabolic rate. Okay, so as your body temperature goes up, you increase the rate with which you burn your fuel. The ingestion of food gives us about a 10 to 20% increase in basal metabolic rate. And age has a major impact on it, because what happens as we age is that cells and tissues die. And as cells and tissues die, their energy needs diminish, and as a result, we don't need as much nutrients, we don't need as many macronutrients, and our rate at which we generate heat from the food that we consume goes down. Now that's by design in a sense, because that allows the population as a whole to survive. If we didn't have our metabolic rate slow down as we aged, if our metabolic rate increased as we got older, what would happen is that those individuals least capable of procuring nourishment and performing useful work would consume the bulk of the community's resources. And what would happen then is that those individuals most able to do useful work and procure nourishment would be selected against. Okay, and so in primitive cultures, this was an important biochemical characteristic that was selected for, which is the diminishing of the basal metabolic rate as we age. Okay. What else affects it? Gender, pregnancy, climate, sleep, and malnutrition are all affected. What happens as we produce heat? Well, we lose a lot of it. Okay, your body has to be continuously breaking down these high energy molecules, and this constantly produces heat. We lose heat through radiation, which is body heat lost to the air around us, through conduction, which is the loss of body heat to the surface, such as when you sit on a cold metal seat, or through convection, which is the loss of body heat to air moving from a fan or from a breeze outside a shower curtain. Just a couple of examples. We also lose it through evaporation. Remember that sweating is one of the major ways we cool the body off. As water converts from a liquid state to a gaseous state, it takes kinetic energy with it. That has the effect of cooling the skin, which cools the blood, which is near the skin's surface, and that cools the body temperature in total. Okay, this also prevents us from overheating, for instance, when we exert ourselves, right? Because if the body temperature gets too high or too low, what happens? Well, we die, right? If the body temperature gets too low,
chemical reactions will slow down. Rule of thumb is that chemical reactions slow down by half for every 10-degree decrease in temperature. And as a result, if we get too cold, what will happen is chemical reactions will slow down or stop. And so we won't be able to do any biosynthesis or any catabolism or anything, and we'll just die. Everything will just slow and shut down. How about body temperature getting too high? Well, if body temperature gets too high, what happens is that the components in the body that mediate chemical reactions will begin to denature. And when they do, the reactions that they facilitate will stop. And that'll also result eventually in the death of cells, the death of tissue, and the death here of the organism. And so what you're looking at here are just some mechanisms that we use to mediate body temperature, right? We see changes in body temperature, which we then counterbalance with a combination of neurological, hormonal, and musculoskeletal responses. And this ultimately resets our body temperature back within homeostatic limits. And thank goodness that that happens, or else we'd be in a bad way. Okay, this brings us to an end of chapter 27. So I will see you guys in a little bit.